I'm going to talk about biology stuff in the deep Earth. And so my background picture for my title slide is actually in the volcanic arc of the Andes. My Argentinian and Chilean colleagues invited us down to study this. But really, most of my talk is going to be about oceans. But hopefully, you'll see how that connects back to the volcanoes by the time I'm done. So what planet is this? Yeah. It's great to flip the Earth around and check out the Pacific Ocean every now and then, because we spend so much time looking at it from the sort of front side. You can almost get a view of Earth that has no land in it. New Zealand creeps in. It's hard to get it out of there. But it's a nice reminder that, we probably all know that percentage of 74% of the Earth is oceans. Well, it really is. If you flip it around, you can remind yourself of that. So since most of the Earth is oceans, if you want to know what's inside Earth, like how life goes into Earth, how deep it goes, then really what you're talking about is marine sediments. That's the majority of the crust area that's habitable by microbes. So we've done some estimates, and there are probably 3 times 10 to the 29 living microbial cells buried in the seafloor. And I know numbers that big are really hard to conceptualize, so that's about 1/3 of the microbes on the planet are buried underneath the seafloor. And it's roughly 10,000 times more than the number of stars in the universe. So this is just a vast, vast ecosystem. So if you want to study life on Earth, you have to look inside Earth, because that's where it is. So how do we do this? How do we go about learning about these deep subsurface microbes? Well, the big thing is that we go out in nature and we get samples. Occasionally people will ask me, like, certainly, you're a scientist sitting at your computer or sitting in the lab bench, and somebody else, some adventurer, goes and gets stuff for you and brings it. I'm like, no, that's the point. I could be doing other fields of science, but I do this one because I get to go to cool places. So we go out, we get samples, and we measure all the chemicals around the microbes. We measure as many things as we can to try to get at what they're eating and breathing, like just how life works in this strange place. And the big thing that we do, usually we do this back in the laboratory, is that we chemically extract their DNA. We pull their DNA out and other biomolecules directly from the natural sample, and this is what we use to infer what they're doing. And then we can also bring home these samples and try to grow things in our laboratories. So the work that I'm going to tell you about right now was taken from oceanic sediments. So we take this ship out. This is the JOIDES Resolution. You can see the big drill stack up on the top of it. This is a long series of, you have to load up pipes. And so the pipes get all taken down to the seafloor. And there's a little explosion that happens at the seafloor, and they get shoved down into the sediment. So this advanced piston coring can bring you back hundreds and hundreds of meters into the Earth, sediments coming back on ship. It's very, very exciting, fun stuff to do. So we go out on ships and we get this stuff, and if you want to start thinking about what kind of microbes are out there, one thing that you sort of have to know as background is that we actually already know quite a bit about the microbes elsewhere on Earth. So one thing we know for sure is that there are a lot of microbes on this planet. So I just found this picture on the internet, and I thought it was really beautiful. It may seem a little disgusting because somebody just put their hand down on a petri dish, and these are the microbes that were on their hand. But if you can think about the fact that that person who made that handprint probably didn't die from an infection from all those microbes. So most of the microbes on the planet do not kill us. So we spend a lot of time in microbiology studying pathogens and diseases, but for the most part, the microbes on Earth are not pathogens. They don't kill us. Although I do want to make the point that we have only been on this Earth for a few million years. Microbes have been here for literally billions of years. So they maybe just haven't figured out how to kill us yet. So I'm going to put a yet on there. It could be. All right, so when we got the first samples that we could be sure were not contaminated, that were really good samples from this deep subsurface environment, they were from off the coast of Peru. And I was lucky enough, I didn't do this work alone. No scientist ever works alone. If somebody tells you, like, so-and-so discovered something, like, no, so-and-so and a bunch of other people discovered things. So this was my first project as a PhD student. So it was a pretty heady time. We were discovering big stuff. And I will show you the data that we found, And I will show you not microbial names or a phylogenetic tree or something really technical. I'll show you each microbe by a box. So I'll put like a rectangle like this up. For every time we got a strain, we got a DNA sequence out from a microbe that it was similar to what somebody had discovered before, if it's dark blue. And I'll make a heat map where the light color is totally different, a DNA sequence that we got that was something that was an entirely new type of microbe that nobody had ever discovered before, something really, really new. So this is what we got. We got almost all white boxes, or pretty light-colored boxes. It was really, truly amazing as an entry into this field to be like, there's this whole undiscovered world down there. There's crazy stuff happening. So as a microbiologist, you look at a result like this and you say, everything there is new. It's time to start culturing things and get things growing in laboratories. So luckily, we had some of the best culturing labs in the world on this ship. And they took samples back to their labs and started growing things. And I'll show you their results, too. And every time they got a microbe that is what we found with our DNA sequences, I'll put it directly underneath the box so they match up. And then new ones, I'll pack to the right of them. And they'll be colored the same way. So if they're light, they're something really new. And if they're dark blue, they're something really old. OK, you ready to see what they found? Zero. Zero of the things that we found, they managed to grow. Nothing. No overlap. And furthermore, everything that they got was very similar to kind of stuff we already knew about. So we realized we had a big problem here. And there was actually another lab that did this as well, and they got a similar result. And I don't want to malign these labs. I mean, they know what they're doing. It's just simply that these microbes were living in a way that we were not adequately replicating. And I know a lot of times people say, well, pressure, it's all because of pressure. And these experiments were not done under pressure, but that has been done quite a bit, and it's not the missing link. You can't just apply pressure to these things and have them grow perfectly. So this is now such a pervasive problem, and we know this all over the world right now, and a lot of samples all over the world. We've actually, a lot of people refer to these things as microbial dark matter. I don't know if you've heard that word, but it's sort of borrowing from the astronomers. But most of the matter in the universe is this dark matter that we know it exists because of its gravitational pull, but we don't really know what it is. So most of the microbes on this Earth are this weird stuff, and we don't really know what it is either. So that's why we sometimes call it microbial dark matter. But the fact that this stuff exists really means that we have to crank back the clock a little bit and talk about microbiology 101, which is, we know, we teach this in classes, microbes grow in cultures, like test tubes, and we have a general conception of what microbes do on this planet. These are sort of like basic tenets of microbiology. But I want to point out that these weird microbes do this to that. That's a framework. It's not necessarily true. These are basic assumptions that our whole field has been based on, but we don't even think of them as assumptions because they're just, well, that's true. But maybe they're not true. This stuff is really making us question that. I think it's important to be able to see your frames. They've allowed us to do that. So if we want to try to figure out, well, what do they do? Let's be aware of our frame of thinking and step out of it. And let's think about this from the microbe's perspective. Get in their head. What's it like to be buried in the seafloor? Well, this is how I think of things. So this is my schematic of seawater overlying sediment. As microbes are buried, it happens very slowly. Sometimes they get kicked back up, and then on average, they are buried and buried and buried slowly, slowly, slowly. The best things to breathe are breathed up very quickly. Oxygen is a fabulous thing to breathe. The fact that we can all be these big multicellular organisms like literally spewing out heat everywhere is so inefficient. We can do that just because we breathe oxygen, which has so much energy. But that gets used up in some places like that far. So that's gone. And then your next things to breathe, like sulfate or iron or manganese, they get breathed up next. And then after that's all gone, the last thing you've got to breathe is carbon dioxide, which is crazy to think of using that as something to breathe. But these microbes can actually do that. But it means that there's a decreasing oxidation potential. So the things that you can get energy from are getting crappier and crappier as you get buried. And you combine this with the fact that the food that you eat, the carbon that you use to make your biomass, where you get your electrons from, that doesn't get replenished. So as they get buried, everything gets worse and worse and worse there too. You have to eat the leftovers of what got laid down sometimes thousands of years previously. So this all adds up to massively decreasing energy availability as you go down in marine sediments. And what we predict is that the number of microbial cells in these sediments should decrease with this. And that's one thing that we actually have measured pretty well around the world. And these are the data. This is a compilation of data from all around the world. And this is actually a form of the plot that I will show more, so try to remember it. Up at the surface, that's the seawater, basically. That's the seafloor. So as you go down into marine mud, on the y-axis here, we have the total number of cells. It's decreasing. So pretty much everywhere in all different oceans around the world, the number of cells are decreasing with depth, which is what we expect. But what we want to know is, like, how fast do they grow? Like, you know, on average, these populations are decreasing. But say if you see a patch of grass one summer, and then you see the same patch of grass another summer, but the patch of grass got smaller, it doesn't mean the grass blades didn't grow. It just means that the total population size changed. So the question is, how fast did they grow? So to do that, we can put this on an age model. So if you look at the burial of marine sediments on the y-axis here, we go down to 5,000 years on the y-axis. So this is some work that my colleagues did looking at the respiration rates and the total turnover time of the population. And already at the surface, we've got cell generation times of months, which is really, really slow. If you put E. coli in a test tube, it will double every 30 minutes. But these things are taking months to double, but that's at the surface. If you fill in the rest of it, they go up to sometimes 50 years or 70 years. So these are microbial cells that don't divide for 50 years, which is weird. Biology is not really designed to deal with that sort of problem, yet that's how we find ourselves. So we have to go to it. So they're not really dividing. They're not really making new cells at all because they don't even have to divide in every 50 years. They could just be switching out their internal biomolecules and fixing the parts that are broken. This is just a total carbon turnover. They don't actually have to make a daughter cell. So it could be that an individual cell hangs out for thousands of years or hundreds of thousands of years. And the thing is that, like, maybe that sounds crazy to all of us, and it is crazy, but it's not. This is actually the consensus in my field. We just say, oh yeah, these cells are probably hundreds of thousands of years old, living cells. So what this means is that my graduate student, as he was learning this stuff, he was really shocked as I was to learn this. And so he came up with a word that I love, so I'm going to share it with you guys. He says, Karen, it's not a biome, it's a diome. I was like, that's so good. That's great. But at this point in the talk, I should probably raise the thing that maybe some of you are thinking, like, why am I doing this? Like, you know, this is an entire ecosystem that's dying. Like, and I'm just devoting my entire professional life to studying this. Not just me, but other people too. Like, what's the point? They're just dying, let them die. But I'll tell you the reason, and the reason is just that I find it utterly fascinating. I mean, how does a cell live for that long? What does that mean? Like, they're still alive. Like, this is a mode of biology that is fundamentally different than all other biology that I've ever heard about. And I don't know how the evolutionary pressures work over such long timescales. I think when you take the rules off of biology, suddenly really strange things can emerge. Like, for instance, if you think about what it takes to get ahead in Darwinian evolution, fast growth makes a ton of sense, right? So when we talk about competition in ecology, we normally talk about the goal is increasing your population size. So we have different flavors of how to win a race, right? You can be the hare, and you can just sprint and get there fast. But we know that being a tortoise is actually also a totally valid way to do to get ahead, and you can still win a race as a tortoise. In ecology, we call these R strategists and K strategists. And K strategists will sort of wait until the rabbits burn off, and then they sort of plod along and they catch up. But all of that ecology is based on growth, increasing your population size. What about this group of microbes who are not actually increasing at all? So maybe I'm proposing that the goal in the deep marine sediments is just existing. So like a Zen sort of thing. You know, you look at a Buddhist monk who's meditating, and he may not look like he's doing anything, but he is. He's making forward progress, even though he's not racing. And if you're not into the religious references, maybe you'll get this reference. For years, I just did not understand the point of this movie, The Big Lebowski. And then I realized there is no point to this movie. The dude abides. So I think that's what these microbes are doing. They're just looking for a white Russian. So in this ecosystem where this seems to be the goal, is just kind of hanging out, what does that look like? How do they do this? So now I'll tell you some, sorry to go from pop reference straight to really technical stuff, but you guys can handle it, right? We did a project in the Baltic Sea, which is the sea that's in between Sweden and Finland and Latvia and Estonia. These are my two amazing students who did all the work on it. We're working on this. This is actually not the JOIDES resolution. We worked on a drill ship that was retrofitted by the Europeans. The ship I showed you before is the American ship. The Japanese have a ship, and then the Europeans make their own ships as needed. So we don't have a ton of ships for this kind of thing. But we wanted to ask the question, what are the potential mechanisms that allow bacteria and archaea, these are two different types of these microbes, to persist in near zero growth state for 8,000 years, which is 50 meters deep into the Baltic Sea sediments. And we did this. I got to tell you about the technique that we used to get these things, because the situation in marine sediments is that there's a big group of microbes. They're a heterogeneous group of lots of different microbes, all bundled together. Of course, they're tiny, they're microbes. And so we need to pick apart individual genomes from these things, but we can't culture them. So we have to pull out a genome from an individual microbe in a mixed population, in literal muck. Like, just think of dirty, crappy muck. That's what we're working with. So what we do is we physically pull out a single cell from this weird, mucky stuff. We do this with flow cytometry, which is just makes tiny little droplets of water. Each droplet has one cell in it, which we pass by two lasers to look at the diffraction patterns as the light scatters off of this thing. So this lets us know roughly the size and shape of what's sitting in the water droplet. And we say, OK, well, that thing looks like a cell. Flick it into its own well. And so in this way, we get a tube that only has one cell in it, which is not much to work with. And then we have to crack it open. We do this usually with changing the pH. We'll like shoot it with some alkaline stuff, because that's fully reversible. Then we apply an enzyme that got pulled out of a virus years ago by some amazing researchers. And we do this to amplify its genomes. We make lots and lots of copies of that one cell. And then we send it off to a magical sequencing machine. And I say magical because it's all proprietary. I don't really know how they work. But they come back with the sequences of DNA. So lengths of DNA. This is tons and tons of data that we get from this. And then we bioinformatically knit it back together into a complete genome after sequencing it broken up. And this is a really fun area of research, putting together data like this. So in this way, we analyze the genes that are present in a microbe. And you can imagine genes just kind of like a menu for what the microbe can do. OK, so when we first got these genomes out of these deep subsurface organisms, we looked for sort of what's abundant, what looks interesting. And something that popped out right away was a bunch of toxin-antitoxin systems. So these are, I'm just showing the data here where on the x-axis I have all the different microbes that we caught. None of these things belong to a phylum that's ever been grown. So these are all this microbial dark matter, strange stuff. And then we got this number of different toxin-antitoxin systems. OK. You probably have not heard of toxin-antitoxin systems, but you should know that evolution has worked out such that we have a lot of cells will make a toxin that kills themselves and then simultaneously produces the antidote to it, and that's why they don't die. Doesn't that sound like a terrible system? But that's how biology works. There's reasons for it. It's kind of a nice, it's like a governor on your system. So you can have this toxin that just sort of slows down your metabolism, makes the cell a little bit sick. And in this way, this is actually used in Mycobacterium tuberculosis. So this is showing the number of, this is the same plot just from a published paper showing how many different of these toxin-antitoxin systems are present in mycoplasma. And this is a horrible disease, obviously, tuberculosis. And one reason why tuberculosis is so pernicious is that if you hit it with a bunch of antibiotics, that destroyed a growing cell, it just stops growing. It's just like, I'm just going to persist and hang out until your body forms this granuloma around it, and then you can't touch it, and you've got tuberculosis for your whole life. So the Mycobacterium tuberculosis uses these toxin-antitoxin systems to get in this low zero growth state. So we have lots of other things that we've identified, but I'm just telling you about one because it would take all day. We think that one of the things these microbes do in the deep subsurface is that they're using toxin systems to sort of slow down their growth rate and be very, very slow. So that's kind of cool. That means that we've known that everyone in this community, in this ecosystem, is starving because the food is 8,000 years old. But that's okay, because they're not trying to grow fast anyway, and they're doing this with toxin-antitoxin systems, among other things. But can we identify what they actually eat? Like, what does one eat when one is living for so long without much food? And to do this, we looked at the metabolites. So that's another sort of technical thing, but basically all the metabolisms that are happening inside cells occur by changing molecules, so individual monomeric molecules. So if you just get a view of all those molecules that are present in the cell at once, that's called a metabolome. And people do this, so my colleagues Sean Campagna and Hector Castro and Eric Tague do this in known organisms, and they get these like subway-looking map things where you can look at the it's like pipelines or dials. So you can look at the upregulation and the downregulation of all these different metabolites. And this is something that people have done in cultures, but we're working in sediments. And it just seemed like a big mess. I mean, it's already a mess in a living organism. And we're going to take this natural sample that's full of, I don't know, tree leaves or whatever dead stuff and try to pick apart individual molecules from it. It just seemed intractable to me. How much do I know? But so the only reason why we ended up doing this work is that guy Hector over there would sort of, whenever he saw me, was like, Karen, you got to put some of your weird deep sea mud in the ICM SMS and let's just look at it. Let's just see what the metabolites look like. And I was like, that just seems like not a great idea, but all right, fine, whatever. So we did it. We put all this stuff in. And I said, so how'd it work out? Did you get anything? And he's like, nah, it was total crap. It just didn't work at all. And I was like, well, obviously. Nothing ever works in the deep sea. That's a truism. And he's like, we only got, out of the thousands that they normally get, he's like, we only got 20 molecules. It's like, 20? That is so much more than 0. Like 20, I can work with 20. And it was great. So my students and I went through those 20 molecules and tried to figure out if we could see what they were important for. And I'll just tell you about one of them, and that's this. It's a molecule that I had never heard of before. It's called allantoin. It's a nitrogen-rich molecule. And what was interesting about what stuck out to us about it is that this is another heat map showing depth on the x-axis here, looking at 2 different cores. So you can see it goes down quite deep. This is down to, I think we've got them down to 100 meters almost into the seafloor. And we got allantoin present in almost every depth in both of these cores, and also another form of it called allantoate. So this is actually a potential food substrate. So this is something that's the product of the degradation of everybody else dying. This is something that would result from that. And it's also a substrate that we never would have thought that these things are capable, like, we wouldn't have guessed that they could eat this. So luckily, we have all these genes to look for. It's like, well, are they eating it? Let's see. So we looked across all the different major groups of microbes that we have. So each column on the left side in the blue-gray stuff is a different microbial group. And then the rows here are different enzymes that are important for allantoin metabolism. And just one guy, not all of them, just this one called Atrobacteria, seemed to be able to eat this allantoin. And so it looked like this one clade, this one type of microbe, kind of had a leg up on everybody else. It was able to eat this food that nothing else could eat, just genetically. So that's what we found, is that it looked like they had a corner on the market for allantoin. So this really brings up the question, if Atrobacteria are the best at eating all the food, why don't they just kill everybody else and win the game? I mean, that makes perfect sense. You've got an inside track to a food source that nothing else has. So just divide your cells a bunch and take everybody over and be the king of the thing. So I'm going to answer that. So we put together its whole metabolism from these genes. So this is a hefty reconstruction of just all the genetic machinery that we could put together, which, obviously you're not going to go into depth and read all this stuff. But it looked like they were a fully functional microbe. They have all the things that you need to make a microbe go. And furthermore, we found that they had all of the genes necessary to make amino acids, to basically just produce this essential building block for all of the proteins that they need. So it looked like what they were doing with this food source was producing some biomass. So that's good. That means that they should divide. So to answer this question of why they weren't just growing like crazy, I went to some more colleagues at Texas A&M and University of Southern California who made some metatranscriptomes out of this. And that is basically just looking at how many of the genes are turned on in the system. It still is amazing to me that they were able to do this in the muck that's deep underneath the Baltic Sea, but they were. So from this list of RNA, or genes that are turned on, that are active and doing stuff, the second most abundant one out of all the genes that could have been there in this metabolism, or in the cellular metabolism, was this one at the bottom that I've circled in blue. And it's an exporter of amino acids. Amino acids are very energetically rich. They're a food source, they're a biomass source, but instead of hanging on to this stuff, it looked like, at least genetically and expression-wise, that this organism was actually giving it away to its friends. I don't know. And I do not like apocalyptic movies because I am scared of movies, but you maybe like apocalyptic movies. I've only seen one of them, though. It was Zombieland. And I actually liked it. I thought Zombieland was pretty good. It wasn't too scary. And I learned from Woody Harrelson that the thing to do when your resources are really, really limited is that you got to get guns, and you got to keep people away from your food. You got to keep your Twinkies. And that is our apocalyptic story, the narrative that we tell each other all the time. Resources are slim, gather them up. Keep everybody away. And here we have this ecosystem that's been dying for 8,000 years, and they're literally giving their amino acids away? Like, that doesn't make any sense at all. So what they could be doing is that if they allow their intracellular concentrations of amino acids to build up too much, they'll have to divide. They'll just have too many resources inside the cell. It'll force all their enzymes to go forward, and they'll make competition from themselves. So maybe, and this is a hypothesis, we don't know. This could be totally wrong at this point. But what we think is that they are releasing these amino acids that they make simply because they don't want to divide. They want to keep in that zero growth state because that's the way to persist. And so if they hoarded, then they would grow too fast. So they have to release it. And then this has the secondary benefit of feeding everybody else in the community. And I don't claim altruism on the part of microbes. That's ridiculous. But it is an amazing system because, our apocalyptic movies are about a recent apocalypse. So these people haven't figured out how to be pros. These are professionals at apocalyptic living, post-apocalyptic living. And what they do is share. And that seems to work out for them. So how does all this stuff get set up? I mean, what I've discussed are adaptations to doing nothing. But we know that Darwin said that you had to pass your genes along to the next generation to get them to be selected for. So how does natural selection happen to allow a microbe to persist for this long? How did this system get set up? And I think for that, too, we have to take our heads out of our frame of reference. For that, we have to remember, now these microbes live for thousands of years or longer. So now we have to start saying, are there geological factors, because that's geological time, that could allow them to come back to the surface? So now, don't think about getting buried down in marine sediments. Now we have to think about that oceanic plate that they're sitting on, because plate tectonics starts to matter when you live a really long time. So what happens in oceanic tectonic plates is that eventually they get crushed underneath continental plates, usually continental plates. So this is sort of a schematic of how to think about this. This is true all up and down Central America and South America. This happens offshore. There's a subducting oceanic plate covered by my mucky guys, that get crushed underneath continents. And early in subduction, sort of at this part right there, all this stuff gets squeezed and gets shoved back up. And so it's possible that these microbes are brought back to the surface early in subduction zones. And of course, they don't survive the full going down to the mantle and mixing with mantle and lava and coming back up in volcanoes. But all of that is probably connected. So maybe this is how this connects to volcanoes. Maybe life deep within the Earth is connected across spatial scales and time scales that biologists are normally sort of restricted from thinking about. So we have a new project. I don't have a lot of results to show you, which is fine, because I'm almost done anyway. But I'll just tell you about what we're doing with this project. I put a dot up for every time we have a sample. So we've sampled with my Costa Rican colleagues all through Costa Rica and Panama along this volcanic arc to try to tie these things together. And this is what it looks like when we sample. We trekked in just a few miles into the jungle, and all this stuff right here, this milky stuff, this is me and some of the rest of the team. We just sort of fan out and with our tubes and real cheap looking stuff, because it is cheap, and sample all this stuff. And this milky red stuff right here is all microbial. So there's tons of these deep subsurface microbes that are sort of spewing out of the earth and taking advantage of the fact that there's this great interface with oxygen at the surface of the earth. And this has also led me to, definitely the second most dangerous site I've ever sampled, inside the crater of Poas volcano. I don't know if you've seen this. This is the closest volcano to San Jose in Costa Rica. And my Costa Rican colleagues actually go there quite a bit. But we went because it was pretty quiescent on that day. But this is looking down into it. So we had to hike down here, and it's like really scribbly, scrabbly rock because it's ejecta. It's not like settled stuff. And you can look at, I mean, there's nothing growing here because the thing blows up every now and then. And this is getting steep. This is basically just like a lake full of phreatic water. So this is steam-heated water that's been like steeped in a tea of volcanic gases. So it's literally battery acid. The pH, this is me with a pH meter right there. It was 0.85 that day. It goes down to -2 though. I'm kind of sad. But I got to tell you, it's terrifying to be in that volcano, not just because of the fear of eruptions, but also because this is a picture of me. It's very steep and, as I said, like scree-filled, and the ground is hot. Duh. It's really hot. But you're like, if you fall into battery acid, but you don't want to fall, so you put your hands down, but it's 600 degrees, so you're like, oh crap. So you just kind of like walk along and keep your boots moving. But I will say, I don't know how many volcanoes I'm going to go into in the future because it was pretty quiet that day, so that's why we went in. But a month and a half after we went in, this happened. This is the shoreline I just showed you. That's where I was standing, right there. It's just a reminder that nature does not care about scientists who are sampling it. It will kill you. And we have to be really, really cautious and not be cowboys out there and really take mitigated risks. And it also means we now know the microbes that are present in this place. This is actually the biggest eruption in 60 years, so we were awfully glad to miss it. We did all call each other on the phone. We're like, everybody, nobody's in the volcano, right? But there are microbes that survive this. So this is a place that microbes live, which is crazy, I think. Like, to think that that's a valid ecosystem for microbes who are like, yeah, cool, I'm good with an explosion every now and then. And it really makes you want to extend your thinking of where life is to outside of Earth. So if you ever think about life outside Earth, which I hope that you do, because it's awfully myopic just to focus on our one planet. This is Mars, of course. For me, I mean, obviously I have my biases. It's hard to imagine life on other planets within our solar system, the ones that we know a lot about, in planetary bodies, because they lack the thick atmosphere. It's hard to live at the surface because you have to deal with magnetic assault, you have to deal with UV assault. It's just a rough place to live. But nothing is stopping their subsurfaces from teeming with life. So when I think about life, and where it might be within our solar system, I think about the subsurface. I am not going to think about, what's sitting at the surface and dealing with the sun's rays. I think it's very, very likely that there's stuff especially on Europa or Io or Ganymede. Those are great places, possibly even Mars. So I hope that you guys will continue to get out of your frame of references as well and think about life on other planets, although maybe you were already there. So I will end by thanking my funders, because I think it is amazing that we have a society that values this kind of science, and I'm grateful for that. This is my building at the University of Tennessee in Knoxville. It's like 4 hours east of here, and it's lovely. It's like a slightly dirtier, scruffier version of the glitter of Nashville. So thanks a lot, and I'll take questions.